Hello everyone. Today we'll talk about the notch signaling pathway in hematopoiesis. As we know, the evolutionarily conserved notch signaling pathway functions as a major mediator of cell fate determination during development and also regulates diverse cellular functions including differentiation, proliferation, and survival. In hematopoiesis, the importance of notch signaling has been associated with both normal hematopoiesis and hematological malignancies, such as T-cell acute lymphoblastic leukemia, multiple myeloma. Blood cell production is maintained by a small population of stem cells in the bone marrow. These cells, we call it HSCs, have the ability to self-renew or differentiate. Cells then differentiate to multipotential and lineage-committed progenitors. These cells can't be distinguished morphologically only by the use of clonogenic assays. The final stage of differentiation is when the cells develop their characteristic morphology and are released into the circulation. This is the process of hematopoiesis. During hematopoiesis, notch proteins are expressed at different stages of development. Notch 1 and Notch 2 are expressed in the CD34 positive, NIN-negative precursors in the human bone marrow. The expression of Notch 1 decreases upon maturation of erythroid progenitors into erythroid cells, while the expression of Notch 2 increases with the differentiation. Notch 1 is essential to direct T-cell lineage commitment at the expense of B-cell development. Deletion of Notch 2 caused lack of marginal zone B cells. Genetic inhibition of notch signaling in HSC leads to accumulation of GMP, reduction of erythrocyte progenitors, and inhibition of marginal zone B cell differentiation and T cell differentiation. In mice, deletion of Notch 1 enzymes can cause ectopic B cell development and fail to develop T cells. On the other hand, increased expression of notch can cause ectopic T cell development in the bone marrow and fail to produce B cells. Let's see in details. Studies show expression of notch ligands DECT1, DELTA-like1, and DELTA-like4 are observed in the thymus, which indicates the importance of notch signaling in T-cell development. Now it is proved that notch signaling plays a role in TCR gene rearrangement. Take a part in the decisions between alpha-beta versus gamma-delta lineage and decisions between CD4 versus CD8 lineages. In bone marrow, inactive Notch 1 allow commitment of a common lymphoid precursors, we call it a CLP, toward the B cell lineage. On the other hand, B cells migrate to the spleen, differentiation into either follicular B cells, we call it a FB, or marginal zone B, we call it a MZB cells in a Notch RBPJ dependent manner. Notch signaling pathway has both ligand and receptors as transmembrane proteins expressed on the cell surface. As such, Notch signaling is in many ways an ideal candidate pathway for instructing communication between HSCS and other niches. As it requires cell-to-cell contact for activation, there still are controversial about whether Notch signaling pathway plays a role in the regulation of adult hematopoietic stem cell in physiological conditions. Firstly, Notch ligands and receptors have been detected in the bone marrow niches and in HSCS, respectively, both in mice and humans. Then, when transducting the constitutively active form of Notch1 intracellular diamond, we call it N1-ICD, or the Notch target HERS1 results is an increase in the self-renewal capacity of HSCS. Further to this, activation of Notch signaling through exposure of primitive hematopoietic cells to Notch ligands in vitro, such as JAK1, extracellular delta-like 1 ligand, and soluble JAK1 also promotes stem cell self-renewal and inhibits differentiation. All the evidences point that Notch has a role in HSCS. On the other hand, loss of function approaches have led to the proposal that the Notch pathway is not required in steady-state hematopoiesis, as highlighted below Table 1. From Table 1, we can see that most of studies support that Notch1 or RBP-GK loss of function has no effect on HSPCs. We need further studies to elucidate the role of Notch signaling in HSCS. Thank you for your attention. Next time, we'll talk about the Notch signaling in hematological malignancies.